And for today, we're excited to have Dr. Aaron Rice from Cornell University, who will be talking about his amazing research on fish sounds. And before I hand it over, please note that this seminar will be recorded and put on the Fish Sounds YouTube channel for future viewing. So hi, YouTube. And we should also have time for questions at the end. So with that, please take it away, Aaron.
All right, thank you. Always super exciting to talk to the Fish Sounds community. Like I was telling Audrey, we got a foot of snow. I may lose power if I disappear. I apologize in advance. But if that happens and people have questions, my contact info is in the lower left. So please feel free to reach out with questions, comments, feedback, all that. So without further ado, what I want to do is talk about the evolution and diversity of fish acoustic communication. I came to the fish sounds world relatively late in my career. Like didn't learn until graduate school that fish made sounds, and I thought I knew a lot about fish up until that point. And what's been really exciting is over the past recent years, seeing how much the field has expanded, how the drop in sensor cost has really democratized the field and enabled a broad diversity of practitioners to get into it. And that's really been great for the field, for the science, and certainly for the fish. And so, when we look at fish and we think about this lovely coral reef, we're hearing these different fish doing their thing. There's some Bacillus albicella from Hawaii. And from a fish sounds perspective, there's a lot going on here. Certainly there's the snapping shrimp in the background. You've got a male damselfish advertising for females, trying to get them in and deposit eggs in his nest. And if we look at the visual representation of the soundscape, it looks something like this, where we see, there we are. This is a spectrogram of the sound where we have time on the x-axis and frequency on the y. So it looks, you would read it sort of like you would music. And so we can see that even with a short snippet of sound, there's a lot happening. We're getting, like I said, the snapping shrimp. We got some damselfish calls in the foreground. We got some parrotfish munching on algae and coral in the background. And there's really quite a bit of contextual information that we can get from understanding and listening to fish sounds. And so, it's not surprising that in the field like bioacoustics, we commonly think we approach it from the perspective of birdsong and birds singing with the dawn chorus around the world. Certainly when there's a cicada bloom happening with the advent of cicadas in the summer. Certainly underwater, there's things like humpback whale song, which has catalyzed an awareness since the 1960s and 1970s over the importance of sound in the ocean. But then there is the increasing recognition of things like toadfish that are dominating soundscapes in coastal waters. And 10 or 20 years ago would be the kind of thing where if you told somebody that you worked on fish sounds, you might get some really weird looks. Nowadays, people get it, and they certainly understand the implications of increases in ocean noise pollution. So it has gone from what was a very esoteric topic to what is now sort of readily intuitive for both scientists and non-scientists alike. But even though there's been this recent development in the field of fish sounds and listening to fish has been around for over 2,000 years. So if we think about in the Western quote unquote scientific literature, the first documentation of fish sounds appears with Aristotle, where the sounds from fish caught by fishermen were observed on the shores and on the deck of fishing boats, and certainly tracing this back through the identified species in Historia Anomalium. These were most certainly gurnards and drum in the Mediterranean. And so what we see time and time again is the names of many fish families or fish species are given to them by fishers who understand that these fish are making sounds, and it's only the scientists with hydrophones that are relatively late to the game that are listening to them. So skip forward, 1,500 years or so from Aristotle, we go to Darwin, where Darwin too was aware of the importance of fish sounds, and was one of the first to speculate that, as in other vertebrate species, the sounds that fish are producing are likely involved in sexual selection, courtship, and mate attraction. And so, again, what's worth pointing out, both with Aristotle and with Darwin, we're still 100 years before the invention of the hydrophone. So we're relying on traditional ecological knowledge in the case of indigenous fishing groups, or we're relying on local expert knowledge of fishermen to infer how fish make sounds. And so, we've gotten this glimpse of individuals making sounds and possibly a behavioral role. But what is really exciting in this discipline of fish sounds, and what summarizes a lot of the research I do in my lab, is being able to transcend biological scales and go from organism to ecosystem all through the studies of fish sounds. So we can start at an anatomical or physiological level, where, if you can see my cursor here, we've got a swim bladder with highly specialized muscles that are dedicated to producing sounds. We have the hindbrain. This is a sagittal slice of the hindbrain with some stained areas that happen to be highly specialized motor neurons in the hindbrain dedicated for generating sound. And of course, the fish ear, which is involved in the detection of sound. We can go from sounds at the anatomical level up to this sort of behavioral interaction where you have males calling for females. In this case, a cartoon of a male midshipman fish calling to attract a female into his nest to lay eggs. We can then go up from that dyadic interaction of males and females to start looking at things at the population level and understand where and when are fish populations spawning. This obviously has a direct role into the conservation and fisheries management of fish, which Audrey will talk about in an upcoming seminar. And then we can take multiple populations of fish within the community and look at sound at an ecosystem level. All of this is possible through a single hydrophone, or in some cases, many hydrophones, where we can have, from this single data stream, have all these different inferences of the science of what animals are doing. And so this becomes a really exciting, integrative, tractable system for asking these kinds of biological questions. For those of you that are new to the language of things like passive acoustic monitoring, being able to sort of do something with the sounds collected, how does this actually work? You might start off with one of these various recording devices. These are just little cartoon schematics here of some that are available on the market, or the recording a fish in a tank. All of these will yield some form of a sound recording that then comes back into the lab, and we can take that and run it through a variety of specialized sound analysis software program, whether that's the Raven software package that we've developed in our lab, or various toolboxes in R. You can use a combination of different detection classification algorithms, also known as AI, to be able to identify sounds. And what we're looking for are these species-specific sounds in the spectrogram using a combination of computer vision or machine learning on the sound itself to identify where and when sounds are occurring within that data stream. And from that, we can make inferences over the timing of patterns of fish calling, or the spatial location. And what ends up happening is that, yes, we're interested in the properties of the sound itself, but from a large scale monitoring perspective, it's that metadata associated with the sound that is equally important. So it's, understanding the timestamp on the detection event and the location of the recorder is what we use for looking at these large scale, broad spatial temporal patterns in fish calling activity. So there's a lot of ground to cover here, and I think what's worth reminding people is that we could do an entire graduate level or undergraduate level course over a semester on the topic of fish sounds alone. So it's a little bit daunting to try to fit all of this into about a 45-minute talk. But I think what best encapsulates starting with fish sounds are our lowly friends, the toadfishes. This is a group where anybody who's worked in the field is familiar with toadfish, and this is best described as a taxon where beauty is in the eye of the beholder. They are these sort of cryptic camouflage guys that hide in coastal habitats, in nests. People think they're ugly. I think they're delightful. I will let you decide what you think they look like. but they're all benthic and predatory, and most certainly most of them are making sounds. But for the vast majority of the science, work has focused on three species. So whether that's the plain-fin midshipman along the North American Pacific coast, ranging from California up through British Columbia, or in the Gulf of Mexico and southern Florida, the Gulf toadfish Apsanis beta, and then going from the east coast of Florida all the way up to Cape Cod, and then beyond northward is the oyster toadfish Apsanis tau. And so nearly everything that we know about toadfish and sounds and hearing, which is a lot, comes from these species. But they don't necessarily encapsulate all of the diversity that we're seeing in toadfish. And what's happening because of their cryptic nature, the more time that people are spending collecting fish in habitats, the more that we are describing. And many of the newly discovered species of toadfish have been discovered in the last 10 to 20 years. So that could be things like this splendid toadfish, which is an endemic species in Cozumel, Mexico. This guy right here on the lower left, or this guy Bifax licinia, which was relatively recently discovered from the Red Sea, with these sort of like fleshy bits sticking out from the sides of its face with eye spots. And so you can see quite a bit of morphological variability within toadfish, and it certainly raises the question how many of them are producing sounds. Most of the work in studying toadfish, because they're hard to see in their habitats, is done from things like this, where this is one of our toadfish shelves of specimens within the Cornell Museum of Vertebrates. You could go to one of the larger repositories of toadfish sounds, which would be at the Smithsonian Natural History Museum or the American Museum of Natural History, where there are fish in jars. And these are great for looking at morphological diversity of swim bladders and mussels. But toadfish are relatively amenable to recording in captivity. So here is a leopard toadfish in our lab, Apsanis pardus, sitting underneath a little flower pot, hoping that he would make sound. But they can also be studied on the reef. So here is an example in the upper right of a Gulf toadfish and a little group of oyster toadfish in New York in the lower right. And so if you know what you're looking for, you can find them. But more often than not in the field, you can hear them. And we'll get into more of that later. What's exciting about and the reason why they have been the subject of intense study for so long starts with their internal morphology for sound production and their sonic mechanism. And so the oyster toadfish here, so this is a lovely illustration from the early 1900s showing this heart-shaped swim bladder they have that sits in the thoracic cavity. And on the left and the right sides of this swim bladder, are these fantastic, highly developed, highly specialized sonic muscles that are dedicated to the production of sound. And so Tower, in this monograph from 1908, described that toadfish make a definite sound he described as either an unk or a kunk, that is produced by the contraction and relaxation of these sonic muscles connected with the swim bladder. You can start to see too that the description of sounds with onomatopoeia is also problematic, because what I would describe them as, you may use a different type of language to describe that. You'll see things referred to as the boat whistle. You'll see things described as boops, all sorts of different things going on. From the 1950s. Here's one of the first recordings of oyster toadfish from Marie Poland Fish's work, where here's a power spectral density. And then Tavolga later described the sounds of Apsanis tau and Apsanis beta. And we can see and hear what they sound like. And so if you were in the water, whether that's off the coast of Florida or swimming off of Cape Cod, if you are in the water listening in the summer months, you will most certainly hear this. If you happen to swim over a calling toadfish nest, your entire body will vibrate. Which is really sort of as a listener and observer of fish, is quite the transformative experience. So here we have our sonic muscles and the internal swim bladder, and where does that sort of sit within the toadfish body plan. So here is the lateral view of the plain fin midshipman. And what we have is in the brain, these vocal motor neurons innervate the swim bladder, which is where the sound is generated. And like I showed you early on, if you take a slice of the hindbrain here, we have this really, really dense body of neurons, the referred to as the vocal motor nucleus. This is work done by Andy Bass's lab at Cornell, where we can start to go and understand the sort of hindbrain generation of where acoustic communication is arising, sort of at the neurological level. If we focus on the brain itself, so this is looking top down at a toadfish brain. So here we have the front of the fish on the left and the back of the fish on the right, and we go from the olfactory bulb to the preoptic area and the optic lobes to the midbrain and then the hindbrain back here. Starting in the preoptic area, we start to see evidence for sounds being encoded that then neurons fire, starting with the preoptic area connected to the periaqueductal gray. We have a vocal pre-pacemaker nucleus here, the vocal pacemaker nuclei on the sides, and going to the vocal motor nucleus, and then neurons extending to the swim bladder here. So this is a fairly hardcore neurobiology, but the exciting takeaway message here is that when we look at neurons firing in the midbrain and the hindbrain, and then we measure muscle contractions and the sound waves, there is a one-to-one correspondence between brain activity, the contracting muscles, and the sound itself. And so by listening to a toadfish, because if you look at the sine wave of the sound, that matches the contraction pattern of the muscles, which matches the firing pattern of the motor neurons, which matches the firing pattern of midbrain neurons. You can essentially understand what's happening in the fish's brain just by listening to the sounds that it produces. And so by listening to a giant colony of calling toadfish or midshipmen, we can really start to get an understanding of what's happening at the neurobiological level of calling at a population. And this is all by listening. So we have our midshipmen here. And for our usual suspects that I showed you before, here is what their swim bladders look like. We have the plain fin midshipman on the left, Upsanus beta and Upsanus tau sort of in the middle. And then in 2000, a group in Portugal described the sounds of the swim bladder of the Lusitanian toadfish, Halibatrachus didactylus, with these sort of funky looking swim bladder compared to these more commonly observed heart-shaped swim bladders that we see in the Perichthes and Upsanus. And so this started to raise the question that even though all of these swim bladders have these bilateral sonic muscles, we're starting to see glimpses of morphological diversity within toadfish. And then when I got to Cornell, my mind started to be blown by looking at this freshwater toadfish that we caught or we got through the aquarium trade. And then upon doing some dissection and clearing and staining, we started to look at the swim bladder, and lo and behold, it wasn't this heart-shaped swim bladder. What this guy had was two distinct swim bladders, and then we later found there was a little piece of tissue connecting them. And so it's like, well, this is different, and this is certainly more different than the Lusitanian toadfish, and it's really different from midshipman and oyster toadfish. So what's going on here? And so we tried to get our hands on as many toadfish swim bladders as we could. So these are from the older literature. And then Diego Voss had a dissertation published describing more swim bladder diversity. And then we got some specimens through the American Museum of Natural History, opened them up, looked at swim bladders, and then lo and behold, we're seeing all sorts of variation on these body plans with the swim bladders, going from heart shape in some taxa to more individually distinct swim bladders in others. And it's like, well, what's going on here? So we took this information and then mapped it onto a genus level phylogeny of toadfishes done by Dave Greenfield. And what we began to see is that by plotting these little cartoons of swim bladder shapes on the phylogeny of toadfish, we see this pattern where all of these guys up here have these sort of single heart-shaped swim bladders, and then all these guys down here have varying forms of two semi-connected halves of the swim bladder. And what became really provocative is that if you look at the biogeography of these two different subclades within the toadfish family, these ones with a single swim bladder shape are the New World toadfish found in the Western Hemisphere. And then these ones down here are these toadfish are found in the Old World or the Eastern Hemisphere. Sorry, Western Hemisphere, Eastern Hemisphere. What ends up happening then it raises the question of the evolution of toadfish sounds, where these two very distinct groups have very different swim bladder morphologies, raising the question of the types of sounds that they produce. Um, what ends up happening is that everywhere we end up looking and listening, we hear different sounds from toadfish. So whether this is off the coast of North Carolina and Georgia, here's Upsanis tau, where you can see variation in the fundamental frequency here. Here is 24 hours of the California, or I'm sorry, this is 5 hours of the soundscape of the Pacific coast. And then this is a 6 month spectrogram off the coast of Belize, where we pulled out a recorder trying to listen for grouper, and then we found that the soundscape itself was saturated with the sounds of Batrachoides gilberti, and this was at Turneffe Atoll. And so the soundscape is being dominated by toadfish sounds at scales of minutes to hours to days, and that's really exciting. So some of the work that we've doing recently in our lab over the past couple of years is trying to identify different species of toadfish that had been described but had never been recorded. And so in the left, we had sounds from Sinopus splendidus, the splendid toadfish in Cozumel. Over here, we have Amphicthes cryptocentris, the Bocone toadfish that Erica Statterman had done in Panama with Smithsonian. And then recently, it's not shown here, but work that we did, which led by Joe Iafredi, we got sounds from the Atlantic midshipman Perichthys plectrodon off the coast of Florida. And so all of these new toadfish species that we are looking at, which hadn't been recorded before, are turning out, yes, they produce sounds. And then with our work in Cozumel, what's been exciting is that in listening for the splendid toadfish, and just sort of as an aside, what we ended up doing was worked with local divers and leveraging local expert knowledge on the fish themselves. We put the hydrophone right in front of a fish nest, so the loudest sounds that we were getting we were pretty confident were splendid toadfish. But we were also seeing these other sounds that were not splendid toadfish that are recurring farther off in the distance. And so that could be any one of these other species of toadfish. I showed you Batrachoides gilberti on the bottom from Belize, but then you have Sinopus johnsoni and Sinopus reticulatus, which were also described in the 1970s, but had never been recorded. And in fact, we know nothing about their natural history. These three species of Sinopus are all from the Western Caribbean, and Sinopus reticulatus and Sinopus splendidus are endemic to Cozumel. And so by having fish sort of just described only from specimens and not knowing anything about their life history, we now, through the perspective of sound, have the opportunity to understand aspects of their behavior and ecology in ways that we didn't have information before. So if we start to take this individual focus, we can then start looking at what's happening sort of at that population level and how different fish species are responding to changes in their environment. So here is work that we had done in collaboration with the National Park Service in Florida Bay on Gulf toadfish. And what was really exciting here is, so this is in Everglades National Park, where we had a long-term recording of toadfish. Here we have a 4 month spectrogram here. And all of these distinct little red lines are the fundamental frequencies of calling Gulf toadfish. What we're seeing these daily patterns or sub-daily patterns, and what became interesting here is it's like, well, what's driving this change? Excuse me for 1 second. The joy of snow days is that I have unexpected visitors come while I'm giving a presentation. Back to this. So we're starting to look at these changes in fundamental frequency, and it's like, okay, well, what's going on? Well, the recorder was co-deployed with a water temperature sensor deployed by the Park Service as well. And what we begin to see is that the changes in frequency of toadfish correspond directly to temperature changes in the water due to tidal flux. And so here we have a pretty direct empirical evidence of how over relatively short-term time periods, the frequency of the fish's call is influenced by the surrounding water temperature. So where these toadfish are, we know they're not occurring in isolation. And so then if they are so loud, in fact, how are they interacting with other sonic species? And so from a study that we had done along the Atlantic coast, this is off the coast of Georgia, where we were looking at oyster toadfish sounds, and we wanted to compare them to black drum. And so you can see a short duration spectrogram of the oyster toadfish call here, and then the black drum spectrogram over on the right. And in contrast, they're both about the same duration, a little over half a second. But the black drum has these downward sloping frequency bands, whereas the toadfish's call is doesn't have that same level of frequency modulation. And what's fun about both oyster toadfish and black drum is that they are loud. So here, if you listen to these black drum, sort of that booming noise that you're hearing. So the source level on these guys is 165 decibels. And when you translate the 165 dB reference to 1 micropascal, which is the convention that we use for underwater acoustics, and we translate that roughly to in-air loudness levels, these guys are about as loud as a jackhammer. They're astonishingly loud and probably one of the loudest fish sounds that we've recorded. So these are individual calls that calling it what we refer to as a low call density, where you can start to see spaces in between them, and some are closer and some are farther away. But when these guys really get into spawning mode, it starts to look something like this, where this is the full-blown chorus, about 15 to 20 seconds, that sounds like this. So it's just this absolute booming noise of a giant school of black drum all calling one after the other. And so what this begins to look like sort of in the acoustic scene is that each of these snippets of sound are 30 seconds of recording. So in the upper left, we have black drum, that same chorus just shown at a different grayscale. And on the right, we have toadfish. And toadfish appear to be a little bit more polite in that instead of these calls all happening one on top of each other, like we see in the drum, the oyster toadfish they take their turns calling. And so they have sort of like these straight lines where one fish is calling right after the other. So if this is 30 seconds of sound, here's what 24 hours of sound looks like. So we have midnight down here on the left, and then midnight the following day, or that same day on the right. And here we have this visible black drum chorus going from midnight to about sunrise, and then starting at about sunset and continuing through the night. And you can see that in this 24-hour recording, here are the sounds of passing ships. Yes, we know that ships are loud, and yes, they dominate that short duration soundscape, but they've got nothing on the contribution to the overall soundscape that black drum are providing. And similarly, if we look at oyster toadfish, we go from 30 seconds of sound down to 24 hours of sound. Here is the sustained toadfish chorus where the toadfish are just calling all day. And then this long duration sound event is probably a storm. And then we have these short duration ship passages as vertical bars. And so if this is 24 hours of sound, here's what 5 months of sound looks like. So we have nothing in January, at least at this large scale. And then around March 1st, black drums start chorusing, where we can see the different frequency bands of their calls. We can see this sort of daily pattern with a vertical black bar followed by space, and then the next black bar. So this is called them calling at night. And then at about April, or when the toadfish start and the black drum start to die down, and they continue calling through the spring. What's really exciting about this particular data set was that this was a passive acoustic survey designed to record North Atlantic right whales. And when I came to what was then the bioacoustics research program at Cornell, and had access to all of this underwater sound initially intended to record whales, we found that it was filled with fish. And so there was a huge opportunity to take advantage of these long duration recordings from around the world and really start to understand what were the patterns of fish calling. So this is at a single site off the coast of Jacksonville, Florida. And if we look up and down the Georgia and Florida coasts, here is work done that we had done with two sensors in North Florida and three sensors in about the middle of Georgia. And so if we look at this southernmost unit here, we can see the earliest onset of black drum and toadfish chorusing throughout the spring. It starts a little bit later at the more northern site and then gets later and later the farther north that you go. And so we assume the onset and of the timing of the spawning season of these different species is related to the warming of the water throughout the spring. So this is one season at five different sites. Here is one site across six different seasons, going from 2009 to 2010, all the way up through winter 2014 into spring of 2015. And what we see as we go down is variation in both the onset and the duration of both black drum calling, toadfish calling. Black drum always call first, toadfish always call second, but they vary when they start and how long they're calling for. And so, in seeing this high level of variability year to year, raises the question like, okay, what's happening with respect to water temperature? So, we took some remote sensing temperature data, look at the variation in temperature on an annual basis, and you can see that even on a single day across years, there might be as much as a 10-degree swing in water temperature. We then took that water temperature information, plotted on sort of a gradient going from cool to warm, and then did an overlay with the spectrogram itself. And then again, this is that same spectrogram that I showed you a little while ago, where what we're seeing is how the onset of the black drum calling is correlated with warmer waters earlier in the spring or late winter. However, it's not a simple on-off, and I'll show you more there in a second. And what that begins to look like, sort of in a fairly coarse binary statistical analysis, is we do see, when using logistic regression, a significant difference with chorusing happening when waters are warmer and no chorusing happening when waters are cooler, but it's not as simple as that. The other fun fact that we were observing is that those of you astutely watching the spectrogram itself may have noticed this increase in fundamental frequency. And this, again, is associated with water temperature, where muscles contract faster under warming waters. And so the warmer the water gets, the faster the muscle contracts. And what we saw in both black drum and toadfish is that black drum calls increase by about 3 hertz per increase of 1 degree in water temperature, and toadfish increase their calling in 11 hertz per 1 degree increase in water temperature. So here's been a nice little story of toadfish with a little bit of species diversity and sort of the ecological relevance, both in terms of short and long-term scale. And then it raises the question, well, what's happening across other families of fish? So we know most about sounds in toadfish, but there are plenty of other notable soniferous fish families. But the vast majority of work has been done on toadfish, on drum, damselfish, gobies, cod, and catfish. And most of the work done over the past 20 to 30 years has been done by fish amenable to recording in captivity, and then only relatively recently applied to field recordings. But these 5 or 6 families of fish are a relatively small portion of the overall fish diversity that we have. And so there was this paper that came out of Belgium in Eric Parmentier's lab that started to ask this question at about the same time that we were asking it, that Kieran and Audrey and Francis at the University of Victoria were starting to ask the question of how many fish are out there, how many are making sounds. Certainly it's beyond just the handful of species that we've been observing. And so we took this sort of provocative idea of how many are out there, and then capitalized on a relatively recent, at the time, a phylogeny that was one of the first to sample all fish and map out on a tree, a phylogenetic tree, of how fish are related to each other. And so if you look at going from sturgeon on the left all the way up to guppies on the right, this is every single species or family of fish that's out there. And so we took this level of phylogenetic tree and used it as the basis for understanding patterns of acoustic communication across fishes as an entire class. And so what we did was that you have about 35,000 species of fish going species by species, as Audrey and her team did, was a little bit intimidating to us. And so we're like, well, let's go ahead and simplify things a little bit and just look at only the 500 or so families of fish that are out there rather than the many thousands of species. And so we took the Reboski tree at a species level, reduced it to a family level. And what that starts to look like is something like this, where on the tree we mapped out at a family level where in the literature there were records of sound production. And what we begin to see is that across the phylogenetic tree of fishes is that there's patterns of sound production occurring across the ray fin fishes as a whole. And in fact, it's not confined to one or two clades. It's pretty much all over the tree. And if you take this and then look at a combination, and we classified differently how the data was being inferred or interpreted to support this claim. We broke it up into morphological inference, audio recordings, and then anecdote or qualitative observations that we trusted. And that looks sort of like this, where you can start to see within these different orders and suborders of ray fin fishes, the different types of evidence going to support this argument, and it's really all over the place, which is quite exciting. And in total, at this point, we found 167 sonic families, but this number is most certainly going to go up because we then took these sort of presence absence at a family level, and then did a little bit of looking with some morphological or with some evolutionary modeling to then start to represent and through ancestral state reconstruction, looking at the probability of sound being ancestral to a certain clade. And so this is what the output of that statistical analysis looks like. This representation is a little bit more fun to look at because we have some fish rather than just probabilities. And so the lineages of fishes that you see here are with a high probability of sounds being ancestral are in blue. Those that have a lower probability of sound are in red. But the nuance to this interpretation is that the vast majority of these red groups here haven't actually been recorded before. Here we have a bunch of the deep sea fish. Here we have a bunch of obscure taxa that are sort of confined to various parts of the world. And so what's likely going to be happening is the more people that are out there recording fish, and listening to different fish groups, the more this is going to be filled in. But what we see is that within the this advanced crown group here, sounds are likely ancestral. They're most certainly ancestral within the catfishes, and we have these several major lineages of sound that not only have widespread sound production, but it may be ancestral to that group. But in fish, it appears from this analysis that sound production, the data suggests that sound production has evolved over 30 times in fishes. Again, I think the more we record and the more species that we understand, this number will likely change, but at least provides an interesting estimate of where are we at in terms of understanding the evolution and diversity of fish acoustic communication. And what's fun about this is we can use this evolutionary framework to inform passive acoustic monitoring strategies in the field for fisheries management and conservation, where we can go from species to sound. So let's say you want to record a catfish. Well, we know that the vast majority of catfish are producing sound. So there's a high likelihood that one would be successful in an effort trying to record catfish. Now, there are silent catfish that are out there, but many of the ones, again, here are data gaps where the fish hadn't been recorded. Similarly, if there's a squirrel fish you want to go after or a species of damselfish you want to go after, there's a good chance that they're making sounds, and you can begin to use field-based passive acoustic monitoring methods to understand calling patterns. So what's exciting about what we found and published in our paper and what Audrey and her team published in their paper was this was at about the same time this analysis came out in tetrapods. And a paper by Chen and Weens in 2020 did a similar approach where they mapped the presence or non-presence at a family level of acoustic communication in tetrapods and found that it had likely evolved independently 6 times in tetrapods. Well, fast forward a couple of years with a paper that came out in 2022 that did something similar. But again, the Chen and Wien study was plagued by data gaps. And this group out of Austria then went in and targeted some turtles as well as some really bizarre and obscure reptile groups and found that lo and behold, there's lots of calling activity in here. And the Yorgowicz-Cohen paper then shows that no, sound production didn't evolve independently 6 times in tetrapods. It's likely ancestral to tetrapods. And so what we're seeing in the field itself is some fairly fundamental overhauls of how we think about the evolution and diversity of acoustic communication across vertebrates, and certainly our inference into a changing perspective in fish and importance of fish sounds is going to play an important role in this fairly important component of biology. So even though we have these large-scale representations of sounds across vertebrates and in fishes, there's still lots of unknowns. So, as of this writing, we've got maybe 1,400 to 1,500 species of siniferous fish that have been described. Comparatively few studies with behavioral context relative to the overall fish diversity that's out there. We have few but increasing numbers of studies in the field. So being able to take lab-based recordings and match that with field recordings. And certainly a lot of the places that we have been working as a community have been in areas that are relatively easy to study. Things like coral reefs, tropical rivers, near populated areas. But places like the deep sea, polar regions, caves are largely unexplored. And so there's lots of opportunity for discovery here. The other component of this democratizing field is the emergence of these sound repositories. So, not to diminish the importance of fishsounds.net being in the lower right, you have a bunch of different online libraries with publicly accessible sounds where anybody who does or does not have access to the ability to record fish in the field can hear sounds for themselves from these different species and start to do essentially office-based work looking at different acoustic mechanisms. And so whether you have the ability to go record in the field or even if not, there's still quite a bit that you can do in terms of learning about and listening to different fish. So I have rambled on long enough. Miraculously, the power has stayed on. I want to thank the very talented staff and students from my lab group at Cornell. As always with this kind of intensive field work, we rely on a wide variety of partners and collaborators across different governments, NGOs, and the private sector and different academic institutions. And so this is very much team-based science, and of course, a wide variety of funders that have supported this work. So I will leave it there and happy to take questions.
Thank you so much, Aaron. That was amazing. And I, as a fan of toadfish, was certainly excited to see how much you were able to cover on what I consider grumpy cuties personally. Yeah. So we do have time for questions. If you want, you can either type them in the chat or you can raise your hand and we'll call on you. But feel free to ask any of your fishy related questions. But maybe I can get us started off. I was curious, and I've heard a lot of discussions of this recently. You talked a lot about variation between species in calling and different environmental parameters. But what about differences between individuals and how identifiable different fish might be?
Yeah, so what's to me has been really exciting is seeing some of the basic behavioral ecology work that's been going on, for the past couple of decades. So what I immediately think of is Art Meerberg's work that was done in the 70s on bicolored damselfish, then later continued through the 90s before his passing, is the ability for bicolored damselfish to recognize each other. And what they observed is the dear neighbor effect, where individual males could distinguish each other with who is a neighbor and who's farther away, such that if you're a territorial fish, it doesn't make sense to spend a whole lot of energy chasing away a nearby fish that's going to happen to be there all the time. And so they could distinguish individuality between sounds of those nearby fish. As well as fish that they're not familiar with. And certainly we're seeing aspects of individuality in different toadfish groups, some of the cichlids, certainly some of the catfish that are out there. I know I'm missing a bunch, but I think this is a really provocative area with how individual information is encoded in individual fish sounds, and how both the fish can tell and distinguish individuals, and how we as scientists might find a way to figure out what the fish can hear.
Amazing. Yeah. And I think it's a super exciting avenue to explore for sure. Um, any questions? Sensing a bit of shyness from today. Oh, I see a hand raised. Michael.
How you doing, Aaron? Nice to hear
Good.
you.
Nice to see
Thanks a
you.
lot.
Thanks for joining.
I have some recording of Peritheus that they seem like they're phasing a little bit, and I assumed that they were two males that were kind of phasing against each other. But I'm wondering if there's some evidence that perhaps the two different lobes of the swim bladder might somehow have some kind of phase effect, some time, time correlated phase effect.
Yeah, absolutely. And so that's a great question. And what's worth giving deference to is some of the work that Joe Cisneros has been doing in his lab up at the University of Washington, where work that he and Andy had done show how the swim bladder of midshipmen also inflates as they're calling, and the fish actually start to become positively buoyant within their nests. And so that's introducing a whole bunch of funky dynamics in terms of the biomechanical properties of the swim bladder itself and sound propagation. And so, certainly we know that there are these male-male interactions which could be doing something weird, but then also as the swim bladder gets going within an individual fish, the properties of the sound are also likely changing due to the resonance properties of the bladder itself. And so I think there's a lot of work to be done there. And certainly now with more sophisticated acoustic equipment, the opportunity to really have a much better understanding of sound propagation in the environment. Work that Joe had done with Dave Zeddes showed the ability for female midshipmen to localize sound on the basis of particle motion trajectories. And so there's got to be sort of a spatial interaction of calling toadfish at the population level.
Thanks, I appreciate that.
Very cool stuff. Any other questions? I do have another one in the absence of any. With you sort of touched on in some of your recordings that there was some human-made noises associated either separately from the fish or of similar loudness with the fish sounds you were hearing. And we do have a seminar coming up that'll talk more about noise pollution. But I thought for now, I would love to hear your thoughts on what the effects of noise, human noise pollution could be on all of these fish sounds we're hearing, and what sort of complications maybe it presents to recording them for all these monitoring applications as well.
Yeah, well, I think part of it is the awareness that animals in their environment, whether it's in air or underwater, are responding to noise in different ways. And so, immediately we start to think of things of masking and sort of that acoustic interference with a noise source and a fish that's trying to make a living by calling. But the reality is that we can take an anthropocentric perspective and really start to think about how are we as humans influenced by noise. So in a noisy environment, yes, I may have a difficult time having a conversation with somebody, but my heart rate might go up. I might start to feel a little bit anxious and stressed. My eating patterns might change. And so, even though in biology we try to avoid that anthropocentric perspective, I think it's useful for informing how these variety of organisms in their natural environment might be responding to noise in the form of stress at behavioral or physiological levels. And then fundamentally, the question becomes, well, what does that mean at an ecological and population viability level? And so I think, I have made the argument that, yes, there is a high level of urgency to understand how noise is influencing marine biodiversity. Certainly, marine mammals have taken precedence for that due to their imperiled status and regulatory protections that have been enacted. But from a scientific perspective, I would argue that fish are a great study system to do this on because, the ability to have empirical experimental evidence demonstrating response of animals to noise is not really tractable in a marine mammal, but you can do it in fish. And so there's been a burgeoning body of work showing how fish respond to noise, both acoustically, behaviorally, as well as physiologically, in captive and field-based studies. And I think this is going to have some really profound impact from the conservation and regulatory communities of how we think about what are the consequences of ocean noise on marine life.
Mhm. Yeah, certainly. And it reminds me of a fun paper I found where they looked at the effects of a music festival on Gulf
Mhm.
toadfish, where they ended up getting stressed from some humans having fun on shore. And so yeah, lots of ranges of effects. And I see Veronica typed human-induced rapid environmental change, perhaps related to this topic. So yeah, would be of interest to see where this goes in the future for sure.
Yeah, and I think,
Um,
oh, go ahead.
oh no, please.
Because I was going to say what I think, I'm sure that, you share my enthusiasm for this, but it's been so exciting for, taking what was a fairly niche subfield of either bioacoustics or ichthyology. And no, we're no longer the scientific weirdos in the crowd where it's like, well, no, this is tying into major conservation themes and the fish that we so dearly love and really enjoy listening to play an important role in terms of how we understand ocean conservation.
For sure. And I'm, yeah, very excited to have joined the field at such an exciting time, for sure. Um, are there any other questions? I'll leave a
Yeah, Audrey,
long
I'd love to ask one.
pause.
Aaron,
Yeah, yeah,
fantastic talk. So I know we talked a little bit about this before, and it's something that I think I'll just continue to think about. So I was wondering if you have any additional thoughts on the common ancestor and or multiple evolution events. Like, is there something where there's going to be a common ancestor? Is this, as the tree fills out, then it'll be lost in certain cases and evolve in other ways? Like, it's weird, right? It's either evolving all over the place or it goes quite far back. And so I was wondering if you have any insight in that.
I don't necessarily have insights. I have opinions, because I think part of it is like no doubt there have been fish that have lost the ability to make sounds. Like I'm not saying that every fish makes sound, um, and certainly what we know, and again, here's where toadfish come into play, is that we know that highly specialized sonic muscles are metabolically expensive to maintain, and the really good evidence for that is that when midshipmen aren't calling and they're out of the reproductive season, there's significant atrophy of those sonic muscles. And so when they go into breeding season, there's huge hypertrophy of the sonic muscles. They change their contraction ability, they change their size. And so what that speaks to is that there is definitely a physiological cost of an ability to vocalize. And there may be some cases where if you're a fish in a really noisy environment like a river, it just may not be worth it to have evolved or the strategy of producing sounds. But what I do think is that with our understanding of the evolution of fish, I do think it's fair to say that sound production, acoustic communication, likely played an important role in the early evolution of fish. And I think that it has likely persisted across the diversification of fish. Now, certainly, and I think some supporting argument for that is that one of the secondary morphological characters of the jawed vertebrates, the gnathostomes, first is the jaw. Second is a relatively well-developed ear that we see in modern forms with three semicircular ear canals. Now, the semicircular ear canal is the vestibular sense, not hearing itself per se, but what we do know is the modern form of the ear that we see in extant taxa evolved very early on in vertebrates and has largely remained unchanged across vertebrate evolution over hundreds of millions of years. And so to me, that reinforces this theme of the importance of sound detection and likely sound production across vertebrates. And so I was a little bit disappointed when I first looked at the data that we got of the independent evolutionary events of sound production, where that was not the hypothesis that I was expecting to be supported. But at the same time, I do think that, fish do all sorts of weird things. They can fly, they eat, literally anything and everything. They breathe air. And so we know that fish have this amazing ability for evolutionary novelty. And I have no doubt that sound production has evolved, been lost, and then probably had secondary evolutionary events again. So certainly, I think the great thing about this being highly collaborative team-based science is that with 36,000 species of fish and lots of questions, there's enough to keep all of us extremely busy for the foreseeable future.
Yeah, that's great. I agree as well. I think there's going to be something really neat about filling in that and looking in more detail because it's going to be certain ways of producing sound are conserved, and then we have all these addition add-ons. It's really exciting to see you fill that out in more detail. Just a random note, I think for your probability analysis, it'd be really cool if someone put together, we've talked about this at FishSounds because we obviously do a lot of outreach. There's a really fun most wanted poster there, just based on probabilities. It can be within data, but I think it'd be really fun because we say it, these ones are likely, and then someone has to dive into that data, but having like a neat comment piece on these are the probabilities of these fish ranked. It really gives people something to hunt for, which I think would be fun.
Well, and I think the opportunity there that you raise is that with the engagement of citizen science. So Isla from our group is going to be talking about the WOPAM work with sort of a global community of sound, bringing together scientists and citizen scientists. What the bird citizen science crowd has done. So for the folks training automated detection algorithms for bird recognition through communities like eBird, they'll tell the citizen scientists, hey, we need more examples of this particular species. Go take your iPhone, go take a shotgun mic, take whatever you can get and go record it. And then sure enough, through the eBird platform, a whole bunch of sounds from under-recorded species start, coming in. And so what could be really cool to do through the FishSounds website is, to go with this most wanted thing. And, we sort of set our eyes on a couple lineages of fish and be like, okay, if people are bored, have access to fish, have hydrophones, or you have, a local aquarium shop, go record these fish because the ability to record, species here and species here on the tree may completely overhaul how we think about the diversity of fish sounds. And so I think being able to engage citizen scientists and people that aren't necessarily familiar with the approach on how to do it presents a lot of opportunities for engagement.
Yeah, we should talk more about that because we could put together a page that has like an outline of who would be of interest. I think we'd probably find a lot of people have many of these species recorded too. Like everyone's got these recordings somewhere and no time to publish them. And so I think let's talk more about that because it would be straightforward to get a good understanding of probabilities and then build a page for it and then kind of put it out to the community to see who wants to help fill it in.
Yeah, well, and I think, with the accessibility of the technology, with hydrophones dropping in costs by orders of magnitude, and the International Quiet Ocean Experiment had a call for proposals to build a low-cost hydrophone. You can now build these things for maybe 10 or 20 bucks or buy one off the shelf for $100. So these are no longer the prohibitively expensive scientific instruments that really, I would argue, constrained the field for a very long time. And so now it's much easier to get hydrophones in the hands of people who are interested in this that previously may not have had the resources.
Yeah. And we can grant fund some of that too, right? Like if someone's that keen, I'll send them a hydromoth. Sounds great to me. Yeah. Cool. But exceptional talk.
Thanks.
Amazing. So maybe with that forward-looking, exciting, aspirational note, we'll wrap it up there. So thank you so much again, Aaron, who I see might have just froze on Zoom. So I think that was really good timing with the storm. But thank you so much for joining, everyone. We will have our next seminar in 2 weeks on freshwater soundscapes with Jack Greenhalgh. You can register for that meeting on the FishSounds Educate page, as well as all of our upcoming seminars in the new year as well. And you can find recordings of this and our future seminars will be posted to our FishSounds YouTube channel. So thank you so much again for joining, and hopefully see you in 2 weeks. Bye, everyone.